Okay, are you ready? Let's get started. Hello and welcome to the 10th ZOOMinar in the Proteinopathy series. It's my pleasure to introduce today's speaker, Dr. Martin Muschol from the University of South Florida. Martin obtained a bachelor's degree in math and physics at the University of Regensburg in Germany. He then completed master's, MA and PhD degrees in physics from CUNY, City University of New York. He then became a research associate at the University of Alabama in Huntsville. And then he was a postdoc, and then followed by that, he was a research assistant professor at the University of Pennsylvania, Philadelphia. Following that, he joined as an independent faculty in the Department of Physics at the University of South Florida. There he has been working on exciting research problems, including protein aggregation, phase separation, colloidal models of protein interactions, imaging neuronal activity, and so on. So it's my pleasure to welcome Martin to present his talk. Martin, virtual floor is all yours. Take it away.
Well, first of all, thank you so much, Magdalena and Joan and Rams, for inviting me to participate in this wonderful seminar series. I will talk today. My laser pointer has doubled up on me, so I'm going to stick with the arrow key. I will talk today about the origins, characteristics, and toxicity of two distinct amyloid oligomer species that we have been investigating. But let me start out first by acknowledging the people that actually have done the work. This includes two of my former students, Chamani Nyangoda and Tatiana Miti, and three of my PhD students, Jeremy Barton, who is just about to graduate and the PhD already wants to pop out there, and Kanchana Karunaratne and Nabila Bushra, and also a talented undergraduate student, Fadia Fakre. I also need to acknowledge a whole host of colleagues here at the University of South Florida who have been involved in this project at various levels. And most recently, my collaborator, Wolfgang Heuer, at the Heinrich Heine University in Germany, and his grad student, Philipp Haseke, and several of his colleagues. And one of the models I will talk to you about was also developed in collaboration with Jeremy Schmidt at Kansas State University. So what I would like to talk to you about is, most of you are of course familiar with it, so I'm going to be pretty short about why we think oligomers are an important potential molecular pathogen in amyloid diseases in general. We'll then talk a little bit about the specific case of lysozyme amyloidosis, because a good amount of work that you will see will involve lysozyme. And then I will talk about the origins and characteristics of two distinct amyloid oligomer species that we've been investigating. We have a lot more information about what I, for lack of a better word, will call early stage oligomers, which we have investigated both in lysozyme and in A-beta, and we have a significant amount of data there. And then a more recent addition to our oligomer family, which I call, again, late stage oligomers, and you will see this is really just to distinguish when we find them. And last, I will discuss the cellular toxicities that's associated with these two distinct oligomer species, and conclude with some potential implications some of our findings might have for in vivo oligomer formation and toxicity. So of course you all are familiar with the basics of Alzheimer's disease pathology that was developed or identified really over 100 years ago by Alzheimer from a patient who showed very severe psychotic symptoms. And he was really the one who showed that this had really a physiological basis. So when he looked at that patient's brain at post-mortem, he found severe brain shrinkage. And of course now the very well-known hallmarks of Alzheimer's disease, which are extracellular plaques of A-beta, shown here in brown, and intracellular tangles of tau, shown here in black. And I will just leave it at that, other than to say that this, of course, led eventually to the amyloid cascade hypothesis, that it's really these fibrillar deposits that are at the top of the cascade that leads to Alzheimer's disease. But that was kind of called into question about 20 years ago, when it was found that in toxicity studies, the fibrils themselves, while toxic, weren't the only thing that showed severe toxic effects when exposing brain tissue to these samples. What they identified, Klein and his colleagues identified, was that there was a second small oligomeric component that was even more toxic or just as toxic as the fibrils shown here, where it severely affected the hippocampal cells and killed them. And even in their work then, they showed that there's tissue specificity, so that the cerebellum, which typically is known not to be involved much in Alzheimer's, was actually quite resistant to these toxic effects. And as with the fibrils, it's been shown that oligomers are also an ubiquitous feature of amyloid assembly. So this is shown here for A-beta 40 and 42, where you see these small early stage oligomeric species before the samples turn into fibrils. And you will actually see in my talk that, yes, A-beta 42 forms those fibrils much more readily than A-beta 40. And sure enough, after that, it was found that you find oligomeric species in just about any amyloid protein you start to look for. This is shown here for lysozyme. You will see our oligomeric species here. They very readily, in this case, assemble into these floppy, we call them curvilinear fibrils. They're oftentimes referred to as protofibrils. And eventually you find the standard amyloid fibrils. I prefer to call them rigid fibrils, curvilinear rigid. And you will see why that's, I think, is a better terminology. And this investigation of oligomers made a big step forward when it was found that antibodies that were raised really against the A-beta oligomeric species also recognized oligomeric species in a whole host of other amyloid proteins, including IAPP, and here's lysozyme, which I will talk about in a second. Since then, it's also been shown that you can isolate various variants of these oligomers from the human brain and from various amyloid proteins, amyloid beta or tau shown here, and they also reemerge in mouse models of amyloid diseases. So the two proteins that, as I already indicated, we will talk about is the amyloid beta peptide associated with Alzheimer's disease and lysozyme, which is associated with mutants of lysozyme, point mutants of lysozyme associated with systemic lysozyme amyloidosis.
Martin,
So
yeah. Your cursor is not visible, laser pointer or arrow.
Let me see. Do you see? Do you see 2 or 1?
I see 2, but still better than none.
I only see 1. So where is? I see the one that's on the E right now of my.
How about using an arrow? Try using an arrow.
Try using an arrow. Let me try that. Or let me try. So if I go away with the arrow, let me see arrow option. Automatic. Still 2. Yeah, this is the arrow. Do you see it now?
I don't see it.
Darn.
People don't see it. So maybe we'll go with the laser pointer. So
yeah, I apologize. So the laser pointer you should follow is the one that's currently over the E. I don't know which one which other one you see. I only see this one that sits over the E in this panel on intestinal mucosa.
So people will follow the right side arrow, laser pointer.
Yeah, my apologies. I have no idea how I managed to do this to make 2 of them appear. All
right, carry on. Sorry for the interruption.
Okay, no problem. So you might be a little bit less familiar with human amyloidosis, lysozyme amyloidosis, or called ALIS. I'm going to show you here what happens is if you have one of the disease-associated mutants, of which there's about 6 of them now, lysozyme starts to deform deposits throughout your body, mostly the intestinal organs, shown here for the liver. But it also shows up in the kidneys, and you see here a typical picture that shows these deposits stained with Congo red and the birefringence that's associated with it. Here in the glomerula, you see the images of these very dense mats of fibrils that are formed, and even in the intestinal mucosa, you see these deposits. Now, one of the surprising results that came out was that the original assumption was if you have one of those point mutations, it would result in a change in the structure of the protein, and this would make it prone to amyloid formation. However, when the structures of two amyloid-associated mutants were solved, you see here an overlay of the wild type and the mutant, and you will have a hard time telling them apart. The wild type is in dark and the mutant is in gray. They were actually structurally almost identical. The main difference was that you had a somewhat higher flexibility of these mutants, and they tended to have a lower thermal stability. But even then, if you look under physiological conditions where these mutants became unstable, this is 50 degrees, so you're still way away from physiological conditions you would expect, or temperatures you would expect these things to become fibrillar. And we will actually come back to this question of flexibility later in the talk. So a lot of the work you're going to see with lysozyme from my lab is done with hen-egg-white lysozyme, which is also very closely related to human lysozyme. It's a 14.3 kilodalton protein, has one amino acid less than the human variant. It's a mixed alpha helical and beta sheet protein. It has only about 60% sequence homology with human lysozyme, but is structurally almost identical to it, and also contains 5 of the 6 amino acids that have been involved with disease-related mutants. And it is a very stable protein. It has 4 disulfide bonds, so this is kind of a brick of a protein, which is one of the reasons why when you try to grow amyloid from it, you have to really work on it. You put this protein above 50 degrees, roughly. Many people put it at 60, and at pH 2 to get it to form these amyloid aggregates. Now, let me go to the center part of my presentation, and that is amyloid oligomers have really been shown to be an important aspect of these diseases. And the interesting part is we really don't understand all that much about them. They're typically considered to be on-pathway intermediates, but our data show actually that much more often they actually seem to be off-pathway competitors of fibril formation. There are also suggestions that they can be fibril dissociation products or maybe fibril catalyzed byproducts of fibril assembly. We don't really understand what are the properties of the protein and or the environmental conditions that are conducive to oligomer formation, and particularly if they're off pathway, how these are different from the ones that promote fibril formation. In terms of the characteristics, they seem to be coming in various sizes and different morphologies. We have only very limited knowledge of really the structure of these oligomers, and there are good indications that just like fibrils, they're also polymorphic. In addition, as I will show you today, there are clear signs that they're interacting with fibrils, and as we have heard in previous talks, they can self-propagate and cross-seed. Finally, the question is, if we have all these various forms of oligomers, which ones are the most physiologically relevant ones? Are they all toxic, or is there some kind of a strain specificity, as I would call it, as for lack of a better word? And then, if that is the case, can we also get some insights? What are the actually structural determinants of this toxicity? So these are some of the questions that we're interested in. And this journey into the oligomer world really started for us by looking at the kinetics of fibril assembly. And when we looked in with our model protein lysozyme, we noticed something quite curious. You can follow, of course, the kinetics of fibril formation with the well-known amyloid dye THT, and we found the characteristic sigmoidal kinetics under one set of conditions. But when we switched or increased the salt concentration of our system above a specific threshold for this protein concentration, we saw a curious switch in the kinetics to what we call biphasic behavior. And we tried to understand what this signified. So when you look in your solutions at different time points in sigmoidal growth, in the lag phase, you really do not find any significant populations of anything other than monomers. there must be some fibril seeds there, but they're so weakly populated, you do not pick them out. However, once you are on this fast rising period, you get this autocatalytic enhancement of fibril formation, and you see increasing numbers and fibrils of increasing lengths as you go along this curve until you reach saturation. That's very different once you enter the biphasic regime. The biphasic regime, you basically lose this lag period, and what's happening in here is you pick up these oligomeric species very early on. They tend to polymerize into these curvilinear fibrils, and once you get the second upswing, you start to see fibrillar species emerging, and as you keep on waiting, these fibrillar species replace the curvilinear fibrils and become the dominant amyloid species at the end. This already tells you that whatever has formed here is metastable, meaning at the end point, if you wait long enough, you will always end up with the fibril species. It also tells you already that it doesn't seem like these oligomeric species formed here are a prerequisite species of amyloid formation. So is this only occurring in lysozyme? And the answer is no. In fact, it was originally observed, biphasic kinetics, and now has been observed by several labs in A-beta 40. But most important for us, my colleague Wolfgang Heuer approached us that they had done kinetics with a dimeric construct of A-beta 40 and had observed exactly the same type of kinetics. So in orange, you always see sigmoidal, and in blue, you see the biphasic kinetics. And sure enough, when they looked in their early stage biphasic region, they ended up with oligomeric species. And once they entered the second phase, they ended up with fibrillar species. We have now shown that the same type of kinetics occurs in A-beta 40 and A-beta 42. And in A-beta 40, we have also clear indications that this follows exactly the same sequence. A-beta 42 is a little trickier because that phase, as you can see here, is actually quite short. And it's maybe even worth pointing out that a lot of the data that have been collected to look at the kinetics of fibril assembly, the nucleated polymerization, were only collected in this range. You don't see them being collected in this range. So do we have some information that these are actually amyloidogenic aggregates? And one way to test that is that you look with infrared spectroscopy. And in infrared spectroscopy, it's well known that amyloid aggregates are characterized by this peak in the amyloid band between 1610 and 1630 wave numbers. And sure enough, our, as I call them, the rigid fibrils or standard amyloid fibrils have a peak there, but so do have our oligomers and curvilinear fibrils. And even, as you will see in a little bit, we can also induce precipitates, even those were already amyloidogenic. Notice that they do, however, differ in their peak positions, so they seem to be structurally distinct. And there is indication that you all tend to pick up a weak intensity around 1680 to 1690 that has been suggested to be associated with antiparallel beta sheet organization. So our presumption at this point is that our rigid fibrils have the traditional cross beta sheet structure, shown here for insulin bundle of fibrils, but here's a single fibril showing this typical cross beta sheet structure. And our oligomers might have something more similar to a cross antiparallel beta barrel structure. So this is actually from the work of David Eisenberg, where he looked at a short peptide and found this barrel structure. Okay, but we wanted to understand, why do we see this rapid transition from what essentially looked like oligomer-free amyloid fibril assembly to this oligomer-dominated fibril assembly? To do so, we actually started to map out the systematics of this transition using mostly THT kinetics, but supporting it also with static light scattering and dynamic light scattering. In both cases, they have different signatures for these two pathways, and also with AFM. And there's also a precipitation pathway, but I'm not going to talk much about that. So you can now vary protein concentration, vary salt concentration, and see if there's some systematic to this change in where this transition from sigmoidal to biphasic growth occurs. And these are our data points here. You see, you need a whole bunch of them to really map out that phase space. And what you find is that there's a whole region either at low protein concentration or low protein concentration or low salt concentration that kind of has this hyperbolic boundary in between, where you only see sigmoidal growth, no sign of these early stage oligomeric species emerging. And then you cross this threshold and there they are. So this was kind of a curious behavior, and we tried to understand, could we understand this protein and salt dependence for the emerging of these oligomeric species? So I'm going to ignore the precipitation boundary because it's not going to play a role anymore. But I will switch to the model that we came up with to explain it. And it is basically borrowed from charged lipid micelle formation. And the idea is that basically we have our monomers, and they have to assemble themselves into an oligomeric species. We assumed that the cohesion that forces that hold this oligomer together are fixed and independent of the environmental conditions. However, the effects of charge repulsion vary with salt, and the effects of entropy that's trying to pull them apart vary with protein concentration. So you can now solve the Poisson-Boltzmann equation and integrate how much energy do you need to pull this together to form this oligomeric species as a function of protein and salt concentration, which is exactly what our phase diagram contained. So these are our data points in the purple squares. And the line here is the fit with this model through the data. And the only adjustable parameter you really have is the cohesion force that we fixed to match the data. And it's actually a surprisingly good model, and it explains why you have this rapid onset of oligomer formation. It's a highly cooperative process that looks to the eye like a phase transition, even though it isn't. So this was a nice way to explain how you get this threshold concentration for oligomer formation. But there was something else really curious about it. There's kinetics. So in orange, again, we have the kinetics where we're in the sigmoidal regime. And as you expect, you see that the lag phases decrease as you increase protein concentration. And in fact, this should follow some sort of power law. The specifics of the power law coefficient is a completely different story, but it should definitely decrease as you increase protein concentration. However, when we entered our biphasic regime, this didn't seem to hold up anymore. So what we did is we analyzed the sigmoidal regime and the biphasic regime to extract the lag periods that occurred under these two conditions. In the sigmoidal regime, we could just use the standard analytical solution to nucleated polymerization and fit our data very nicely. And then you extract the threshold, you fix the threshold for when you decide this nucleated. In the biphasic regime, we had to be a bit smarter, but we only cared about the initial stages. So we said this is dominated by oligomer formation at first. So let's subtract that by assuming it's an nth order irreversible assembly process, fit that to our kinetics, and then subtract these nth order kinetics to recover the underlying fiable kinetics. And that's shown here. And now we're in the same process where we can fit those curves and extract a lag period. So what do we get? Below our critical oligomer concentration, we indeed see that our lag phase decreases as we increase lysozyme concentration, as you would expect, and it follows a nice power law behavior. Once you cross the critical oligomer concentration, you actually start to see a turnaround, and the oligomers now start to actually inhibit fibril formation. This is, to me, a clear indication that they cannot be on pathway. They have to be off pathway because they actually interfere and inhibit fibril formation. It's curious to notice that the same observation qualitatively was made by Susan Lindquist when she originally proposed the nucleated polymerization model, that she said it didn't quite make sense that the more oligomers she had, the less likely it was that she nucleated her fibrils. I think they saw exactly the same thing in their system as well. And we have shown this now, Wolfgang Hoyer has shown that the same thing is true for A beta in his system. So how do we understand that? Why do they do this? And our assumption was that it has actually to do with an interaction between the oligomeric species and curvilinear fibrils with the fibril seeds, that they actually suppress the process of secondary nucleation. And to test that, we isolated fibrils under sigmoidal growth conditions. We isolated the curvilinear fibrils and oligomers during the early stages of biphasic growth, and we imaged them in AFM. Then we mixed them together and saw what do we see once they've been mixed together. And sure enough, now we very frequently or consistently see that these oligomers like to associate themselves with the fibrils. This is here shown for A beta 40. This is shown here for the dimeric construct of A beta 40. Again, fibrils, oligomers, put them together. And here for lysozyme. And lysozyme was even more curious because we could see when we mixed our oligomers in with the fibrils, you saw some association. But when you heated them to the growth temperature, remember, we need 52 degrees, not just did they associate, they actually caused the fibrils to bundle. And again, we did a control where we just heated the fibrils. They didn't do that by themselves. It's only in the presence of these oligomeric species that they actually started to bundle. So it actually acts like a glue, pushing them together and thereby interfering with fiber growth. And this is the kinetic analysis from Wolfgang's lab, where they also looked at what happens if we add in our amyloid oligomer species and try to interfere with the oligomers process directly by seeding them in. The trick here was that he used the oligomers from A beta, the dimeric construct of A beta, and that allowed him to preserve them under conditions where A beta 40 by itself wouldn't grow these oligomers. So he could add them in, and sure enough, as he increased the amount of oligomers he added in, he increased the lag period. And that's shown here, that if you just add monomeric, the monomeric version of the dimeric construct, you have no visible effect. However, if you put the oligomeric species in, you see this nice decrease. Fitting it to the kinetics, he could also show that it wasn't consistent, this effect was not consistent with an effect on primary nucleation, but consistent with secondary nucleation interference. So the model is that basically this secondary fibril-mediated secondary nucleation process is suppressed by having these oligomers bind to the fibrils and thereby taking up the sites that would be required for fibril-mediated secondary nucleation. So what do we know about these early stage oligomers so far? We know that they're present both in A beta 40, A beta 42, in the dimeric construct of A beta, and in N-acquired lysozyme. And that's actually quite remarkable if you think about it, because one is clearly an IDP, and the other one is a natively folded protein. Nevertheless, you find all these commonalities in the assembly process into the fibril state. We know that they're metastable aggregates. They are their formation is confined above some critical oligomer concentration that's distinct from the threshold for fibril formation. So they're not obligatory fibril precursors. You could form fibrils day in and day out and never encounter these oligomeric species. A colloidal model readily replicates this critical oligomer concentration behavior. We know that these oligomeric species are structurally distinct from RFs, from the rigid fibrils. They're off-pathway competitors to fibril formation, and they actively interact with and inhibit fibril formation. So we were quite pleased, and we thought we had made some very nice headway, and probably made the mistake to continue and do additional experiments, particularly toxicity experiments. And as I will show you towards the end, these toxicity experiments, when we looked at the toxicity of fibrils, we isolated our fibrils, and as a control, we ran whatever was not in the fibril. Basically, we isolated them, spun them down, and used the supernatant of the fibril as control to show the supernatant had nothing. Unfortunately, the supernatant had clearly something in it that was highly toxic, which was really not what we had expected. And it was surprising because remember I showed you that under these conditions, when we looked with AFM, we never picked up anything in there, right? So somehow it was out there at the end of the growth process. And the question was how, first of all, what are these things that are highly toxic? And second of all, when do they emerge and how do they emerge? And so we call them late stage oligomers mostly because that's when we found them. They occur at the late stages of sigmoidal fiber growth. So the first thing we wanted to make sure is that somehow, since we centrifuge our fibrils to separate them, could we have induced an artifact by breaking our fibrils during centrifugation, right? And so we have like some short fibril fragments that causes toxicity. We didn't see anything in AFM on our supernatant that suggested, or TM that suggested that. And similarly in light scattering, we just saw a peak in our supernatant that didn't show any kind of larger aggregates in there. And it's actually, light scattering is very sensitive to the presence of even minuscule amount of large aggregates. But then we ran it over a size exclusion column, our supernatant, and sure enough, there we found an oligomeric peak in addition to the monomer peak that we pick up when we run monomeric lysozyme or even hydrolyzed monomeric lysozyme of our column. So clearly there was an oligomeric species in there. So if you look at these various aggregate populations we then had, was that we had rigid fibrils that we could grow under sigmoidal growth. And that's kind of a typical TM of what they look like at pH 2. We could grow our early stage oligomers, and which very, very readily actually polymerize into curvilinear fibrils. It's actually hard, particularly for lysozyme, to not have them polymerize on you. And these late stage oligomers. So these are AFMs of the isolated late stage oligomers. They don't seem to form very fast. It's kind of interesting because that's something Rakesh Kayat had mentioned about tau oligomers, that some of them do polymerize into protofibrils and some do not. And we seem to see the same behavior here. So we wanted to figure out where and how do these oligomers form, right? So we didn't want to dismiss the possibility that maybe they had formed there and they were THD positive and somehow we had missed them after all. There is a case like that in IAPP where apparently there is THD negative oligomers that form in the lag phase and cause toxicity. The other option that we liked better was that maybe these oligomeric species emerge as a dissociation product of rigid fibrils, or that rigid fibrils catalyze the formation of these late stage oligomers by interacting with monomers. Particularly, this thesis had some nice ideas because it would immediately tie into the polymorphism of fibrils. If you have polymorphs of fibrils and they dissociate and shed oligomers, you would have different oligomeric species, right? And fibril polymorphism would tie to oligomer polymorphism. So we like this idea. And as many ideas that you like a lot, the data then hit you over the head and say you're completely wrong. So what we did to test when do these things emerge, we had to first be able to separate them. Remember, our protocol for separating them is that we have to spin down our fibrils and then we show what's in the supernatant. So that's what we basically did. But we did it in two somewhat different ways. So we set up a whole series of solutions and then disrupted or stopped the growth at different time points, which is shown here. But we set it up in two ways. One was the way we grow these aggregates for our toxicity assays, which is in centrifuge tubes because we need a significant amount of volume. But we knew from experience that when we grow them in 96 well plates, the fibril formation is accelerated. And that is because they have a smaller volume, and we're quite confident that what happens is that the air-water interface actually promotes fibril formation. So it accelerates the fibril growth process. So you actually grow fibrils faster in the smaller volume than in the large volume. So we could see how the emergence of these late-stage oligomers correlated with the emergence of the fibrils. So in black, we couldn't really easily measure our fibril component here, but we knew from experience that we have to wait about 4 days to have fibrils emerge in our centrifuge tubes. And in black, you see the amount of oligomers we detected in the supernatant. And sure enough, as we usually expect fibrils to emerge, we also saw the emergence of these oligomeric species. However, when we now monitored fibril formation in our 96 well plate, as expected, we saw a much faster and much earlier upswing in fibril growth. Yet, our late-stage oligomers were delayed in formation. So instead of correlating with fibril formation, they anti-correlate with fibril formation. They actually compete with fibril formation. Again, a very strong indication that they're off pathway from fibril growth. And in fact, we directly had originally tested whether fibrils can shed these oligomers. After we had isolated them, nothing came off them. And we also checked whether, when we added monomers back in, whether the fibrils could catalyze this oligomer formation for 2 days, because that's the lag. Remember the lag period, there was nothing that appears here. So we can for 2 days, if we throw in fibrils and see the fresh solution, we know we're not gonna get spontaneous late stage oligomer formation. Nothing happened. So again, something else was responsible for the formation of these off pathway oligomers. And what we finally settled on was what could cause this delay in oligomer formation. And we realized that, of course, one thing that's been well known in this field is that lysozyme also gets hydrolyzed as a function of time, particularly at these very acidic pHs. So our slight worry was, what we were dealing with is that we generated a new fragment of lysozyme, and that was the one that was responsible for this new oligomeric species. To test this, we ran reducing SDS and looked at what's in the soup surrounding, the monomeric soup surrounding our oligomers versus what are our oligomers composed of. Because if a fragment would show up, it should accumulate in these oligomeric species. So this is shown here with reducing SDS pH. If we use just fresh monomers, there's nothing there. In fact, these bands are actually an artifact of heating your solutions at 90 degrees with SDS. You induce some hydrolysis there. And that's true for the early stage oligomers and their monomers. They basically look identical. This remains true for the late stage oligomers and late stage oligomers here and their monomers. So what that means is that these oligomeric species are still formed by the whole length protein, but it is severely hydrolyzed and held together by these very strong disulfide bonds. So it's basically a frazzled version of the monomeric protein that does it. How about immunoreactivity? So this has caused us some headache, but in fact, if you look in the literature, there's 1 or 2 reports where they tried to do this and didn't succeed. We did succeed to get A11 reactivity. And what we found is that our rigid fibrils have some non-specific reactivity. And we think this is due to the fact that they actually form plaques on these surfaces. And we have evidence for that. And these are extremely sticky, and they just, anything sticks to these plaques when you put them down. In fact, we had to reduce the fibril component here to get equal concentrations of protein to deposit on the fibril, on our membranes. Our early stage oligomers, very little response. Clearly, very significant response to the late stage oligomers. And in the question and answer session, I can tell you, it depends a little bit which A11 variant you use, which reactivity you get. But this is a commercial one that we use, and it shows very, very strong response to the late stage oligomeric species, not the monomers that we isolated from the same time point, and not fresh monomers either. You can also do some sort of tinctorial characterization. So these are 3 fibril dyes, and we show the response to the fibrils, but also to these 2 species of early stage and late stage oligomers. And as expected, they have weak responses, but they're clearly distinct for these 2 oligomer species. And then we tested 2 oligomeric specific dyes that are considered to be much more sensitive to oligomers. One of them is bis-ANS, and the other one is one we identified ourselves, crystal violet. And again, these 2 oligomer species have clearly distinct tinctorial characteristics as well. So you could say that also the tinctorial profiles for these three populations, rigid fibrils, early stage oligomers, and late stage oligomers, are all distinct. So what do we know about these late stage oligomers so far? We know that in the case of lysozyme, they require partial hydrolysis. They do not form unless your protein is significantly, has significant levels of partial hydrolysis. They're again off pathway from the rigid fibrils. They're structurally distinct. We have some FTIR data and the dye response profiles I just showed you. We think they're composed of dimers or trimers, maybe even a tetramer, but they're definitely smaller than our early stage oligomers, which happen to be, in our case, octomers. They don't polymerize into these curvilinear fibrils, and they have a strong reactivity with an oligomer antibody. So this brings us to the cell toxicity studies, which actually kind of alerted us to the presence of the secondary oligomer species. And that brought up another issue that I think is oftentimes overlooked in the field. We grow our amyloid species at pH 2. So our naive assumption was when we throw them on the cells, well, things kind of might change slightly, but basically we have what we have. Well, unfortunately, that's not true. Once you bring it over to pH 7, your fibrils form plaques. Your early stage oligomers and curvilinear fibrils form these open gel-like clusters. And these guys we couldn't even image because they form such large precipitates also that the grids, the TEM grids, would just break. But we can look at them under the microscope. So if we look with light microscopy, in bright field, you see these very well-defined fibrillar plaques, have beautiful THS fluorescence, and they readily can be seen under cross polarizers, no Congo red staining required. Our early stage oligomers and curvilinear fibrils form these open, smaller gel clusters, and our late stage oligomers form these kind of sheet-like large aggregates. However, they do not show any internal organization as is betrayed by the cross polarization. So they're not fibrillar in structure. It's also probably worth pointing out that particularly for the RFs, or our rigid fibrils, of course, that's very close to what you would see in the lytic plaques. Whereas our early stage oligomers have the open structure that's typically associated with diffuse plaques. And so I think that's also kind of interesting to realize that maybe this provides us some insights of what's the origin of these different types of features that's been seen in pathology, and as far as I know, have never been clearly identified of what they really represent. But let me go to our toxicity studies, and I have to say, this is kind of still work somewhat in progress. Overall, we're very confident that we have it right now, but the numbers might still slightly change. So we ran a whole bunch of different controls. What you see is that the rigid fibrils show some toxicity, but actually this will probably change somewhat, because that's also a word of caution. It probably depends to what extent they have assembled into the plaques or not. The more plaques you have, the less toxic they become. So we suspect under some circumstances, you have soluble fibrillar species that are actually toxic, and the plaques are probably quite inert. So this might still change somewhat. To our surprise, our early stage oligomers showed very little toxicity in our whole cell imaging assay. I think Wolfgang has to say something else about this, because when he tried these species that he generated with A-beta, they did find synaptic deficits in nerve cells, but also not whole cell toxicity. So it's very interesting that, you know, depending on how you run these assays, you have to be very careful of how you interpret your results. And finally, this is actually not even the final version yet. This was before we actually had started to isolate our late stage oligomer species. This was really just our supernatant. And notice how really severely toxic these late stage oligomers are in our system. But we also had another benefit of using an imaging assay compared to the standard MTT assay that doesn't do that. When we looked in the microscope at our various controls, of course we had our peptide control, our toxic peptide control, and the cells get really obliterated. But there wasn't, other than kind of some overall loss of cell numbers, there wasn't all that much you saw in terms of what rigid fibrils or early stage oligomers did to your cell culture. This changed completely when you added these late stage oligomers. They literally obliterated the cell layers and altered the morphology of the cells dramatically. So not just did it affect the individual cells, it also disrupted tissue structure. And this, I think, is actually quite interesting because that's, of course, what's observed in many amyloid diseases, might be related to vascular AD, the disruption of endothelial cell layers in blood vessels. And we even see that in kind of like, you might call this almost like a spongiform type pathology. That's quite a stretch, and I'm not going to push that too much, but I think it is really interesting, and I think it's worth looking at what your cells look like when you expose them to these different types of aggregates. Okay, so let me sum up. I'm actually kind of overstaying my welcome here otherwise. Clearly, we have shown that you can generate polymorphs of oligomers under in vitro growth conditions that are actually pretty much identical. And we've heard before that such polymorphism has been found post-mortem in patients as well and in animal models. We think that maybe different oligomer polymorphs might very well have very distinct biological activity, as our data suggest. Right? We have found some insights into what actually allows different oligomeric species to emerge. We had found this COC for our early stage oligomers, and I think the hydrolysis mostly is also interesting to show that maybe the structural flexibility of your protein is a critical factor determining under what type of oligomeric species it can form. So it's kind of like the conformational spectrum of your protein that might play an important role. The assembly of our aggregates into higher order structures also seems to affect cell toxicity. So we have to actually be careful what we saw cell toxicity. Are these higher order assemblies that are toxic, or are the original oligomeric species that we found in solution the toxic species? I think that also needs to be looked at much more carefully. And finally, I think it suggests that the picture of what amyloid diseases involve is a lot more complex, that it really involves a whole cascade of distinct amyloid aggregate species. They can interact among themselves within a specific protein. They can evolve over time and change, therefore, probably their biological activity. And as we have heard from Rakesh Kayat and others, they can interact with other amyloid proteins in your system and affect their formation. And with that, I want to again highlight the people that actually have done a lot of the work, and thank you, and I look forward to the questions.
Thank you very much for the great talk, Martin. Really appreciate it. So let's look at, there must be a lot of questions in the Q&A, or you can raise your hands to come and directly ask the question to the speaker. Let me start the question to you, Martin.
Mm-hmm.
So it looks like most of your detection of oligomers relied on THT. In many cases, THT doesn't work. It doesn't detect the oligomer formation. So did you ever come across some other oligomers which are not detectable by THT?
Let me put it this way. Our oligomers we didn't first find our oligomers, our late stage oligomers, because they're basically obliterated by the THT response of the fibrils. The THT of the response of the fibrils completely covers them. We personally haven't found some that are not responsive, but this could be, protein specific. I mean, the case of IAPP is a common one. Right, you had reported that you saw some oligomers that were not, and so I definitely would not exclude it. And again, it might be dependent on the structure of these oligomers.
Absolutely.
Right.
Yeah,
So,
we have seen the CMC like behavior, micells like behavior for IAPP.
right.
We detected using pyrene dye, so it wasn't sensitive to THT, and we had to do a lot of NMR to prove that as well. So Vipin, go ahead and ask the question.
Thanks, Martin, for a very nice talk. I'm very curious about your curvilinear fibrils, which sort of seem to shuttle between the oligomer stage and back to the curvilinear fibrils. So what is the fundamental structural difference that you think between the rigid fibrils versus the curvilinear fibrils? morphologically, sure, they're different, but there must be some sort of an organization on a molecular level, right?
This is what I would say. What I suspect is that the building blocks, we know that the building blocks of these curvilinear fibers are the oligomers, not the individual monomers, right? And we've shown that previously with AFM that the heights of the oligomers matches the heights of these, the curvilinear fibers matches the heights of the oligomers, but not of the fibrils. But this is the model we have in mind, and that's the best we can do so far. We think that's really the right model for the fibrils is basically the steric zipper, and cross beta sheet structure. For the oligomers, I suspect, and this is really purely hypothesis at this point, that maybe this structure actually is a good one, that they start actually forming these polymers by attaching themselves here at the ends. So you would have this as your growth unit, and the protofibrils would kind of form by attaching themselves at the end and polymerizing. And that might explain why you actually also see them still as individual identities in the curvilinear fibrils. They show up as really kind of beads on a string. That's the best we have so far.
Okay. Thanks. So Anna, I have two more questions related to the curvilinear fibrils. One is more of a practical question.
Mm-hmm.
You seem to have somehow isolated or got a pure form of the, it wasn't an ensemble of different species, but you had clear pictures of curvilinear fibrils.
Mm-hmm.
And I was wondering, is it just the protein concentration versus the salt variation that you use to sort of isolate these or get it in a pure form?
I would almost say the opposite. I wish I could isolate our oligomers and not have them constantly form these darn curvilinear fibrils, so we could get a better sense of what the oligomers in the early stage pathway do versus what the curvilinear fibrils do, because we have a very hard time not have them polymerized immediately into these curvilinear fibrils. They very rapidly try to do so. And so that's also when you get the best yield of it. So the problem is we don't have trouble forming the curvilinear fibrils. We have trouble keeping the early stage oligomers from polymerizing on us. It's almost that kind of problem. It's almost the reverse. Does that answer your question?
Yeah, sort of. I mean, there are some species that we also see curvilinear fibrils, and they seem to be stable forever. So that's the reason I was asking.
Yeah, have you tried seeding late stage fibrils into them? Because when we do that, what we have found, they are metastable. So it takes much longer to get rid of these curvilinear fibrils than it takes to grow fibrils without them, right? Yes. So then, in fact, you see that in our kinetics as well. Where was here? Yeah. Right? You see they flatten out. This actually means your reaction, our growth reaction isn't at the end of 96 hours. We had to wait go out there about 2 weeks before all the curvilinear fibrils had disappeared and had been replaced by rigid fibrils. So they have different levels of metastability, and you might have to wait a long time because the fibrils basically have to depolymerize these curvilinear fibrils. So you have to pull the monomers off them and then add them to your fibril state. That's a very, very slow process.
Yeah, 5 weeks and counting. So
yeah. And depending on what type of, how metastable your oligomeric species are, it might even take longer than that. It becomes, when we do it under the wrong conditions, these things gel out. The curvilinear fibrils gel out, and there is no way in hell you're going to ever get them to depolymerize. They just hang around for months.
Thanks, Vipin. Let's take a question from Surajit. Surajit, go ahead. He's not here. Okay, the next person, Srijoni.
Hello, Professor. It was a nice talk. So I was just curious if you can just, or maybe I just missed it, if you can walk through the process of isolating these fibrils. So you said like you need to like kind of centrifuge before you start growing them. So is it like what you do even if you're growing it in a 96 well plate?
No. What I said is we need to get rid of the fibrils to see what was in there. So we grow them always from the start, right? But then we need to separate the fibril background from the oligomers. And the fibrils we can spin down. they're big enough that we can spin them down, and then we can see what's in there other than fibrils. Does that answer your question?
Okay, yeah.
Mm-hmm.
And so another question. So since you're saying that like how these go into like curvilinear fibrils, so is it kind of hard to like grow crystals out of these oligomers?
Polymers are notoriously difficult to crystallize, and they're polymers. And in fact, they're the worst kind. They're floppy polymers.
Okay.
Floppy polymers don't like to crystallize.
Okay.
And typically the crystals you form are not the kind of crystals you expect from small molecules. So we haven't really tried this, and maybe it can be done. I think the microcrystals of the fibrils is much more realistic, and that's also why you see a lot of data coming out of this now. Maybe cryo-EM could get there.
Yeah.
Okay, so Sudipta, go ahead.
Hi, Martin. It was a very nice talk.
Thank you.
I have a question about this, your oligomers being recognized by these common antibody, which recognizes the amyloid beta oligomers, etc.
Can I refuse to answer that?
Well, I have a question and a proposition that we should all probably look into. It seems like, and those oligomers you find are the most toxic ones. So it seems like, would you be suggesting that it's really a structure that is toxic and not the sequence?
I think it is really the structure. That's right. It really depends what kind of structure you form, and that's even shown in toxicity. Look, the sequence doesn't change if I have fibrils or oligomers, yet they show different toxicity, right? And even among different oligomer species, I see different levels of toxicity. So I think it's the structural organization that does seem to matter, at least from what we can tell.
Is that a specific structure? Would you like to go out on a limb and say, some people suggest that this could be beta barrels, which are actually porine-like shapes, which permeate the membrane or something like that? Would you?
I think it's really
these are tetramers. You're saying there may be dimers and trimers, maybe tetramers.
Right.
So it cannot be a very large structure.
Right. So I agree with you that I think it's not quite clear yet. And all I was saying is we see dimers and trimers along the way. Whether that's the structure that causes toxicity would mean you have to be able to separate them and say which one of those is actually the toxic species, right? Which is another complication in the process. What I can say is that when we look, they do have at least different tinctorial profiles. We see differences in the FTIR spectra. All of this suggests that they're structurally distinct, even so they all in FTIR show that they have amyloidogenic characteristics, meaning these intermolecular beta sheet bonds. That seems to be common, but then they seem to be distinct among themselves. And unfortunately, all we have so far is FTIR, which seems to show differences for A beta and lysozyme. We have some tinctorial differences, which I showed you here. And we have antibodies that seem to recognize different variants of it. So to get to your antibody question, we do have another antibody that was given to us that showed that it preferentially recognized the early stage oligomers, not the late stage ones. So again, it suggests there are structural differences and that different antibodies for oligomers recognize different polymorphs. And that's actually also what Rakis Kayes had said for tau, where he can grow these different variants of oligomers and raise antibodies against them.
Okay, thank you, Sudipta. Let's go to Wang. Wang, go ahead.
Dr. Martin, that's a great talk. Thank you so much. And here my question is, you introduce different species to the cell to look at toxicity. Do you ever look at like a similar system like Matt Damon developed like a the biosensor like aggregation, and to compare like what's differences of aggregation purposity of different species? In your thought, which species do you think have a higher ability to induce aggregation in cell, and what's correlation between the aggregation and the cell toxicity in the cell system? Yeah, thank you.
I'm sorry, what was that you had early on? You said, like, have you ever looked at something else? And didn't quite catch it. What was the other thing you wanted us to look at? I didn't catch that.
For example, you like as a McDermott done is if you have a different species like a fibro or oligomers, you can transfer to the biosensor cell or like a cell model to see how this species induce endogenous aggregation in the cell model. Because like people think of fibrillation and aggregation maybe it's two different biological processes. Right. And my question is, if do you ever compare, or how about your thoughts, your different oligomer species and fibrils, which one might have the higher ability to induce aggregation in cell, and how this aggregation correlate to the cell toxic?
Yeah, honestly, we have. It's a very interesting question, and we haven't looked at that. And partially because we're not cell biologists, so even our toxicity assays, we already kind of went out on a limb there. but nevertheless, I thought looking at what happens even in these assays is very important. And I agree with you. For another question is, of course, how do these things proliferate, right? And so you want to understand how do they proliferate within a tissue and spread throughout tissues. And I honestly suspect we will find that the lysozyme amyloidosis is, remember, there is clear indications that the huge amount of fibrils you form have to have some effect on the tissues, right? So it can't just be oligomers, right? And so how do they spread? It's a great question, and we haven't looked at that.
Yeah, I have a second question. Is do you ever look at the tau fibril, whether or not tau will follow like this similar like polymorphism with the tau? And that's the question.
Well, I mean, we know that the Ruckus Kite has shown that tau has various forms of tau oligomer polymorphs, right? We haven't looked at that. What we would like to see next probably is, can we identify something comparable to our late stage oligomers with A-beta, because we already have some nice A-beta data. And so, and the answer is, I don't know yet. We have definitely looked with human lysozyme, because this was all Hennig lysozyme, and we definitely find the late stage oligomers there as well. But that's as far as we have gone.
Okay.
Thank you. Thank you so much.
Can we have a question from Wang?
Sure. Yeah. So hi, Martin.
Hi there. How are you?
I'm fine. Yeah. So for your cell assay, I mean, trying to follow up Long's question. You know, when I look at literature, this happens to different kind of fibroblasts in particular. Did you try the different cell types or different coding or different surface, glass with plastic and so on?
Not yet, no. I think for me, the more interesting one would be, is this really something that's specific to endothelial cells, right? That you disrupt these endothelial cell layers by adding these toxic oligomers. But yeah, we haven't looked at that on the cell biology level at all. No, but that's as far as we've gone.
Yeah, because you know, lysine is a enzyme that degrades sugars. So I bet it will probably organize or change the organization of collagen or some extracellular matrix. So how about the different type, oligomer, fiber? Do they have different effects?
Yes.
Or do they have
in fact,
the
monomers in our case have no effect. So this is actually the native monomer. This is the hydrolyzed monomer. This is the medium. And yeah, these are the different effects of our aggregates. So rigid fibrils do seem to show toxicity, even so, again, it depends probably on the overall stage of assembly. Early stage oligomers in our case, pretty much innocuous, and late stage oligomers, severely toxic.
Okay.
And again, why this is so difficult is because even there we haven't optimized yet, characterizing what it is you actually have present in your soup, right? And I think that's another thing, that it's not just the cell biology that matters, it's also, as we suspect, the type of aggregate that's present and on the cell. And that characterizing that is actually not that straightforward. And it actually has taken us a lot of time even getting to this point, and even this is not yet optimal.
So you're using the one from the company, right?
Which
the lysozyme. Do you
yes.
purify it, or how much glycan is on it?
We don't know, but it's actually we've tested that we can actually, it's multiple times recrystallized. We don't know how much glycan is on it, but again, remember the monomer itself has no effect.
So, okay, glycosylation has no effect on oligomerization?
I can't say that, but we have, they provide the mass spec and there's no indication that there's anything else in there, right?
Yeah, but sugar is a part of the, this is the glycoprotein. So they say it's pure. It doesn't mean there's no sugar on it.
Well, but if you look at the molecular weight, you kind of would know that, right? If there's something else on there, then there should be based on molecular weight.
Yeah, if it shows like a smear-ish, then that means that's sugar.
Right, but I mean, even in our simple analysis here, yeah, you can see the smear, but that's because it's been overstained.
Yeah.
But really, it comes out all the time at a very nice uniform molecular weight. But that's just SDS. That's not exactly high resolution, so I wouldn't say that shows you if there's some sugars on there. But mass spec would.
Okay. Yeah. So I didn't catch exactly how the oligomer is more preferably coupled to the fibers when you co-incubate them.
Sorry, you didn't catch what about the oligomers?
So you co-incubate it, monomer or oligomer with the fibers. Can you explain the mechanism why the oligomer bind to the fiber better than the monomers?
You mean these data where we looked at the... Yeah, okay. So no, what we have here is we isolate the fibrils. We take the monomers out of the equation, right?
Okay. Yeah.
We isolate the fibrils and we isolate the curvilinear fibrils from the monomers, right?
Mm-hmm.
And then we mix them together. So we have them, we either deposit them directly or we first mix them together and then deposit them.
Okay.
And then we look at the deposition. What does it look like? Do these things like to associate themselves? The curvilinear fibrils associate themselves with the fibrils.
Yeah.
And remember, it's only in the presence of these curvilinear fibrils we see the inhibition. We don't see the inhibition when we increase monomers per se, right?
Mm-hmm.
So when we increase monomers per se under the sigmoidal growth conditions, we don't see an effect on lag period of fibrils. It's only once you form these oligomeric species that you see the effect.
So here you could use Mark Diamond's, the FRAT assay. If you label.
We could.
Yeah.
But I think this is pretty convincing too.
Yeah,
right?
yeah, it's good. Yeah.
Right.
Okay.
And the lag period tell you too. And on top of it, my colleague Wolfgang Heuer just had did another experiment because one of the reviewers had said, well, aren't all proteins sticky? So couldn't all proteins do this? So we threw in ferritin, which is a huge protein, and had no effect. And in fact, the Dobson group has also shown when they used various proteins, they don't affect this. You really have to have specific interactions. Not every type of aggregate or every type of protein does it.
Okay, we'll take two last questions before the end of the formal session. Danilo, and then followed by Srijit.
Okay. Hello, Martin.
Hi there, how are you?
Fine. We are fighting against the pandemics.
Well, you're way behind us. We are much better at this, obviously. We have much more infection, much higher death rates. We do better than anybody else.
Well, now we are a bit in trouble right now.
I know.
However, very brilliant talk. My congratulations. I'm very, very curious about the biphasic kinetics. Aggregation of A beta.
Okay.
Because in the past, I observed that I was looking for some inhibitors, of course. And some of the small molecules I was screening, they could induce a biphasic kinetics.
Interesting, okay.
But I discarded them because I considered them as an artifact, and I discarded them.
Yeah.
Now you are inspiring me because I think that it could be worthy to reconsider these small molecules And my question is, what we should expect from these experiments with these small molecules? What they can help us to better understand the formation of these amyloid oligomers. What is your opinion on that?
Well, first of all, I always want to add caution. We really carefully check that the biphasic kinetics we see is associated with oligomer formation. There could be other reasons why you get biphasic behavior. So I wouldn't immediately say just because you see biphasic, you see early stage oligomer formation. However, I have no problem seeing that using small molecule compounds, you might push exactly the system like we have done with salt, right? Remember that we changed when we see oligomers or not by simply changing protein or salt concentration, right? That's shown here, right? You go along here, and if you change salt to a higher level, then that boundary will be at a completely different location, right? So your small molecules might just shift the COC for oligomer formation, and that's what you might have seen, right? That it preferential.
That's what I was thinking.
Right. So the question is that a good thing or a bad thing? And that depends very much, are these globular oligomers and early stage oligomers we see a good thing or a bad thing? If they are a good thing, yeah, you might actually delay the process of fibril formation, because as we have just heard, they can really slow things down dramatically, right? If they're a bad thing, you don't want them hanging around, and you might have just promoted that they're hanging around for even longer. So that depends very much whether we are confident whether these off-pathway oligomers are something that helps or hurts in the evolution of pathology.
Thank you. I have to reconsider the small molecules.
Yeah, no, that's very interesting. That's really neat.
We will be in touch, however.
Oh, that sounds great. I look forward to it.
Thanks, Danilo.
Thanks so much.
Srijit, go ahead. Srijit. Okay, looks like Srijit is not there. Okay, so we are ending the formal session. I'm sorry?
I have one question, Rams.
Go ahead. Why not you ask in the postdoc student session, in the informal session?
All right. Then
thank you very much, Martin, for the great talk, and thank you all for participating.
Thanks for having me.
Coming here. Now we are moving on to the informal session where students and postdocs, you can raise your hands and you can join the panel directly, and you can be interacting with the speakers and others directly. Longcheng, are you a postdoc or student? If not, you already asked a question, so anyway, I'm going to promote you to the panelist. No faculty.
Bikash, did you want to ask a question?
Yeah,
Bikash,
I
go
just
ahead. Yeah.
my question. Very nice talk. I was just wondering, like, as like Professor Danilo and many people like mentioned, we usually see the similar kind of like stuff when we do the experiment in many different kind of conditions and RAMs, and we always discuss, like, we try to figure out actually how the TST data can be very nicely interpreted. So looking at your data, like if you look at A beta 42 and A beta 40, so you have two different parameters. One is like your like concentration in your solution, keeping the volume constant, and we know that A beta 42 aggregates much faster than A beta 40. So which clearly gives an idea that the faster kinetics, like the molecule that aggregate faster, whether you have a parameter of more concentration or properties, that is giving more secondary nucleation, right?
Right.
If I'm correct. So in that case, like, did you also try to look at the secking condition by changing different secking condition at a very low concentration, if you can see those kind of secondary nucleation in your sample?
Secondary nucleation of A beta 40 versus 42?
Yeah, like you have shown a different concentration. For example, in 2 micromolar, you didn't see that. You see in 5 micromolar, 10 micromolar, and 100 micromolar.
Right, exactly. So this follows the COC logic. Let me see, where is it? Actually, I should probably go, well, let me just see if I can quickly get there. Here, it's before that. Sorry about that. That's what you're referring to.
Yeah, like,
so
my question here is that if you look at A beta 40 or A beta 42, let's say 5 micromolar,
right.
if you will sec this sample faster, do you expect that you will see a secondary nucleation here?
This pathway, when I show you the orange traces, they are always dominated actually by secondary nucleation. The reason why you know that is because they have this very strong autocatalytic behavior. If it was primary nucleation that dominates the growth, you would actually start to see a slow drift of fibrils forming consistently, because you basically have this fixed probability. Which that tells you is that the probability of primary nucleation in these systems is miserably small. But once you form them, you get this autocatalytic acceleration of fibril formation, where the formation of a few fibrils accelerates the formation of more fibrils. And that's exactly the signature of secondary nucleation. So yes, all these curves here in orange are dominated by secondary nucleation. Does that answer your question?
Yeah, I think that makes some sense. And is there any like explanation, like I'm expecting, like if you have a higher concentration, so that is also proportional to your precipitation in the sample, because you have 100 microliter or 200 microliter sample volume in your 96 well plate, right? So if you look at the 100 micromolar sample in A beta 40 or like 50 micromolar sample A beta 42, as you have more sample and they are aggregating at a certain stage, they are not more soluble in the solution. They can precipitate, right?
Well, I think what you could have is you form larger aggregates. That would be the only thing that's really relevant, because you have more protein to start with. And so as the aggregates get larger, they might, because you have more monomers to feed them, they might be larger. But I think otherwise it doesn't have that much of an effect, because this mostly happens at the very late stages when you have these very large aggregates. So there, the amount of material you have could matter. But what we found is much more important is that the lag periods shorten dramatically if you actually lower the volume. And we think that has to do with the surface effects, the air-water interface.
What was surprising for me is that, like, when we study about IAPP, we always see that once you see the TST curve goes up, after some time it goes down, because we always believe that because of the precipitation.
That could very well be, yeah.
But in your case, I didn't see that. So that was a little surprising for me. So if you can comment.
Yeah, notice, actually, that's a very good point. And the reason, the difference is, I think, the following, and that is, let me go here. Sorry, I have to do it. Maybe I should just stop. No, I don't want to stop sharing because I want you to see this. You go to the cell experiments here. Aggregates do not bundle much and don't precipitate that much. They can gel out on you. Volume, the whole volume can gel out on you, but they move. Forget that. They immediately come out of solution and precipitate. And I think that's what you're seeing, that you see fibril formation, and as they get to a certain size, they start forming these larger precipitates, and they just settle out on you, and you lose them in your cuvettes. So that pH 2 is nice because it preserves these aggregates in suspension, and that's not at all a given under various conditions.
Very nice explanation.
Yeah, actually, Dr. Martin, that's my question is about this, because I know for your cell toxic, you have this species and made it, then you added the cell culture medium. And the cell culture medium pH should be around like a pH 7 to pH 7.4.
That's right.
And now the different species, they might play the toxic role, extracellular, or they can be intaken by the cell and play the role, toxic role. If they intake in the cell, they might follow the lysosome pathway or endosome pathway. So the pH will be more acidic. So what's your thought about how the species, extracellular, intracellular, or specific cell, like an organelle compartment to play different toxic? Yeah, do you follow that? Yeah.
Yeah, actually, that in many ways, that's really one of the points of this phase diagram, that Sheena Radford has shown with B2M that depending on which pH you use, you end up with, he gets much more oligomer formation of A beta when he pushes it towards the lysosomal pH, right? So yes, these environmental conditions is exactly what I would say are critical for understanding what type of aggregates do you actually use, right? But we don't really fully understand that. And we also, to answer your question, we haven't looked at uptake of these specific aggregates into our cells. We don't know whether any of them get in. Given that they form these very large aggregates and precipitates, I don't think so.
Yeah, because we have same issue with the tau, and we found actually we do the fibrillation, but we are more on the cell biology part. So we're using different concentration for tau monomers and do different shaking, make the fibril, but we didn't look at the EM, the structure. But when we introduce this fibril to the cell, we found actually we can see dose-dependent more aggregation, but beyond some concentration, the aggregation ability is dropped down. So then we raise different possibility. Maybe the high concentration fibril is too long, cannot intake by the cell. Maybe they can intake the cell, but they can lose the decrease the ability. But today your talk is very insightful. We should look at the fibrillation. Maybe on the fibril stage, maybe higher concentration of tau, they cannot develop fibril. Maybe they have more oligomers happening there. We should look at that. That's very, very helpful.
Yeah, and it's actually, I think it really is an issue that hasn't been looked at. And, you know, that's more in our wheel, that to look at these aggregates than the cell biology. So we kind of focused on that. So yeah, because we're just ignorant about cell biology. But I think that's actually a very, very important point. And in fact, we even found one has to be very careful with toxicity assays per se. We only found, for example, with the very dyes that are used for the cell toxicity assays, right? So you might get readout of what you do to the aggregates instead of what the dyes do to the cells. Right. So another little problem.
Okay, thanks a lot. Sahil, go ahead.
Yeah, I have a question. So we have curvilinear fibers, and with time they can go to the rigid fibers, right? In your case, if you you can precipitate the curvilinear fibers. If you precipitate it and then remove the salt, which is the cause for making them the curvilinear, then what will happen? They will go to rigid fibril faster, or
they actually will dissolve. So if you isolate them and you put them under conditions where they haven't formed before, they will start dissolving. And that's actually a sign of their metastability. So that phase boundary I showed you here, I really want to get out of this. I don't want to stop sharing, but I want to get out of the escape. There we go. So we actually had done these experiments. So this is a slide I didn't show you. So if we take these oligomers and we change salt concentration from above the COC to below the COC, above the COC they grow, below the COC they decay.
Okay, but okay, they will decay and become monomer, or then they will start forming the rigid fiber.
Eventually, if you have them under these conditions, you build up enough monomers that they can reform the fibrils. In fact, here this is shown. If you see the fibrils grow under all these conditions. So yes, they can basically depolymerize and therefore form the basis for fibril formation. But we haven't done that experiment, but that's what I would expect. Let me put it this way.
Thanks.
Okay, it looks like, Martin, you answered all the questions. There are no other questions. Anybody else? Okay, here, Long is coming again.
Sure.
Okay. Go ahead, Long. You have to unmute.
Yeah, we can't hear you right now.
Okay, now it's fun. Yeah, because so many questions, so insightful. So, Dr. Martin, and we look at it, 42 and 40. Do you ever look at other firms all over, like the A beta, like CTF, and the longer version for
Actually, what the data that I showed you in the beginning of the biphasic behavior were of a dynamic construct of A beta. That's the one that Wolfgang Heuer had generated, and they actually have a much lower threshold for oligomer formation because you basically already dimerized them, so you basically locally increase the concentration. Let me see if I have this. This graph here, there it is. This is the dimeric version of A beta.
Yeah.
And it has a much lower threshold. Notice at 1.7 micromolar, it already forms oligomers. For A beta 40, it's around 30 or 40 micromolar. For A beta 42, it's between 10 and 20.
Yeah. So here, because some people have the conception like the cell toxic caused by A beta in the 80, actually some is from the APP processing before the A beta 4042 generation. That's a long version, like a cytoplasmic truncation APP. So that's people think the toxic caused by this species rather than A beta 40 and 42. So I'm wondering, and for this version of APP precursor, do they also follow same this dynamic?
I mean, the fact is there are suggestions if your protein gets, or your primary sequence gets too long, it tends to suppress the ability for fibril formation, because it becomes increasingly, entropy basically hits you over the head, that there's enough energy in the hydrogen bonding to overcome the entropy of these very long proteins. But that's the assumption, but that doesn't mean that there couldn't be another fragment that can form that, and I wouldn't know the answer to that.
Okay, thank you so much.
So there's one question from Ella Chini, who left the attendance from here. So I will ask the question, Martin, on his behalf. Let's see here. He's wondering what you think are what you think are the key structural and physical attributes common to toxic and non-toxic oligomers. Are there any similarity or difference?
We thought we had something, but we have to confirm this. And that is the beta barrel, extent of the beta barrel. So you notice that, for example, our oligomer dyes, they had tended to have a stronger response to these late-stage oligomers than to the early-stage oligomers. And what we had seen in FTIR, which we're trying to confirm, but our FTIR went down and hasn't been resurrected lately, that we also saw a stronger anti-parallel beta sheet peak in there. That's the closest we've come to some structural correlate between this. Whether this is what drives it or not, it's really just at most a correlation. And unless you have the structure at hand, that's really hard to do, hard to say.
So this growth incompetent non-toxic oligomers, do they grow in size as a function of time?
Which ones are we talking about?
The early oligomers, which are
the early stage oligomers, they mostly polymerize. So they seem to be stuck at about 8 monomers. And then once they have formed that structure, they should be confined in size. Because if you think about it, just like with a CMC, with a micelle, it's confusions, right? That's how you get to these oligomeric structure or micellar-like structures. And what happens next is that they can start to polymerize. That's how they grow.
Okay, great, thank you. So one last question. Is it okay to take one more question?
Sure, of course. How many you want?
Bin Su. Go ahead, Bin. Bin, you have to unmute and talk. Hello, Bin.
You're still muted, Bin.
Can
there
you
we
hear
go.
Me?
Yeah.
Yep.
Oh, very nice topic as always. Thank you for your data. I have a question related to amyloid and small molecule inhibitor interaction. You may have mentioned about off-pathway aggregates. So there are multiple labs showing in the literature that, for example, interaction of IAPP with EGCG, the work by like Jeff Carey or by Wanker from Germany, showing like a broad range smear like in SDS page. Can you comment on the those? What are those species in terms of physical property, and how can we identify because I think the mechanism is still poorly understood.
I agree. It's poorly understood of what exactly EGCG does. It looks to me that it pushes the system into kind of like a precipitate-like state, right? That's disordered. that where EGCG kind of acts like a glue, and it loses some of the structure. That's why you get the smear, because it's not the well-defined oligomeric structure you had before, right? But that's the closest I could say about that. And with the on pathway versus off pathway, one comment, if you allow me to make, is you have to be very careful when you judge whether something is on pathway or not. Just because it appears early on doesn't mean it's on pathway. It only means it's kinetically favored over the fibrils. Whether that means it's on pathway or not is a completely different question. So I suspect they generated yet another off pathway oligomeric species with their compound.
Okay, thank you very much.
Great, thank you. It's 11:30. Magda, do you have any question?
Yeah, my question's regarding like how you think your off-pathway oligomers interact with the fibrils. You're showing that they, like when you mix the fibrils, they tend to clump together. Yes. Do you have any speculation on the structures, on the mechanistic insights, which portions?
I honestly don't, why they have this very strong affinity for the fibrils, but their fibrils, the answer is I don't know. I don't know why they do it, but they seem to have a strong affinity for the fibrils. How this comes about, maybe it's some hydrophobic interaction, but literally we don't really know.
Is it on the side of the fibrils,
yes.
On the tip of the fibrils?
No, we know it's this, it has to be the side, because the tip would inhibit elongation. If you put it at the tip of the fibril, you disrupt elongation. So if the fact that it interferes with secondary fibril-mediated nucleation means it's on the side of the fibril. And that's what we see here too. Look at how, do you see my cursor there?
Yeah.
How it's just kind of decorating the side of the fibril. And that's also how people think secondary nucleation works, right? That it's fibril-mediated secondary nucleation.
And my second question, you were saying that your off-pathway oligomers, if I understood it correctly, they are mostly proteolized. So you're actually, they're proteolized, that they're like composed of a different fragment, right?
No, actually that was exactly the opposite point I was trying to make.
Okay, so I just got it, yeah.
So no, that probably means I didn't explain it well. What you see here is the level of hydrolysis that the monomers experience at different stages of growth, right? Our early stage oligomers, this is the level we see with the late stage oligomers. If any fragment would also show up as enhanced band in the oligomeric bands, and it doesn't. They look exactly identical. So whatever you have in the soup surrounding the oligomer is also in the oligomer at the same proportion.
Okay, that cancels my second question, because I was thinking that they're like composed of proteolized fibrils.
That's what we were worried about, right? No. That's exactly why we did this experiment.
Okay, yeah. Thank you.
Okay, more than 11:30 now. So thank you very much, Martin. Thank you all very much for the participation. Great, and let's say bye-bye to everybody.
And you guys have a nice weekend, and thanks so much for having me.
Thank you, Martin.
Take care.
Yeah, great talk.
Thanks.
I really enjoyed it.
Thanks.
Thanks.